influenza viruses are just generically prone to recombination because they have a segmented genome. They have 8 segments of RNA that encode all of the proteins. And the original thought was that you can have very distinct recombination events when 2 different strains, say of an influenza A virus, infect the same cell. Then you're producing all of these segments simultaneously as part of viral replication, and they can just randomly resort into progeny virus. This recombination event, which is called antigenic shift, is thought to have led to some of the major pandemics of the 20th century. You think the other source of antigenic diversity, that is variation in the genome that leads to variation in the amino acid sequences of the proteins and therefore eluding the immune system, is the fact that it is an RNA virus, so it uses an RNA-dependent polymerase. And when you use an RNA template as opposed to a DNA template, it's much more prone to error. There's this misconception that in a typical flu year, where you're trying to make a vaccine, you base it on what was circulating at the very end of the last season. But in some years, you get this so-called drifting of the strain so that it changes by the time that you need to vaccinate people, and or even after you vaccinate the people, it changes, and therefore they're not protected. In truth, it's always happening. The virus has this incredible amount of genetic diversity that can undergo natural selection. What's the selection pressure? The selection pressure is the immune system. That's the main. That's one of the major pressures. The virus does not have the ability to persistently infect anybody. It only can survive if it goes from person to person to person. So once you've had the infection and have had an immune response, the only way that virus can infect a subsequent viral strain can infect you again is it has to have a new set of determinants that are not recognized by the immune system. So there's always pressure for it to change. It's constant. So typically what's done is, and this has already been done with the H1N1, is the vaccine, the typical flu vaccine strategy is you focus on the hemagglutinin and neuraminidase, the H and the N, because that's really where the protective antibodies are, if you're going for a vaccine that works in that way. And so you make a recombinant virus, you take an old virus that's been around for a long time called PR834, and you do in vitro what I was saying happens with antigenic drift. You purposefully make a recombinant in which you have all of the internal proteins of the parent virus, the PR834, and only the hemagglutinin and the neuraminidase segments, 2 of the segments, are from the virus of interest. That's already been done. That's not difficult to do. That virus now is sent out to all the vaccine manufacturers. They inoculate into eggs. They then purify the virus, and then they do what's called splitting the virus. So when they split the virus, they take the whole virus and they now treat it with a detergent to get rid of a lot of the internal proteins and enrich for the hemagglutinin and the neuraminidase. But there's some of these residual impurities. So why do they do that? They do that because many years ago, when they used to just use whole inactivated virus, they would take the virus and activate it with formalin, and then that was what was injected. But they found that certain groups had a higher rate of reactions to the vaccine. So this became the standard, at least in most of the developed world, to use a so-called split vaccine. Flu would be in the inactivated category,
Okay.
not subunit. So it's not encoded. So splitting, in a sense, acts to sort of make it subunit-like, but not as pure as if you actually used a recombinant strategy.
Okay.
I do actually have some unpublished stuff. I had my lab send some blood samples. So these were a series of children, 5 to 9 years of age, who normally have pretty good responses to vaccine. And they had gotten what's called the trivalent inactivated vaccine. Excuse me. So this has an H3N2 strain, an H1N1 strain, and a type B influenza strain. So these are the three strains that circulate in various amounts every year. And we had we had blood samples at the baseline and then after vaccination to see how much antibody immunity they got to the vaccine components. And the question is there any cross-reaction between the H1N1 that's circulating now in terms of antibody protection that they got vaccinated with and the swine influenza H1N1? And the answer is, it's kind of shocking me, there's none. We saw nothing. So this H1 is quite immunologically distinct. And there's no question that particularly people that have not been exposed to very much H1N1 natural infection, like younger people, they're going to need young children, the elderly, the immunocompromised, they're all going to need to be vaccinated with a specific vaccine that's targeted to this H1. Older people seem to be doing better with this virus than younger people. Many of the deaths have been in somewhat immunocompromised young people, not in the elderly. We do not have any evidence that this is particularly, it's not following the usual pattern of most of the mortality being in people over 65. But so far, it doesn't look like that. It really does look like it's different. And one idea I have is that there may be some cross-protection from wild-type H1N1 infections, but you may have to go back a ways to see it. It will happen. It has to happen. It will happen. It's a priority of the government. The real question is how many doses. I think that it's going to be kind of like women and children first. Children tend to have higher levels of virus and excrete it longer. And you can show that when in certain countries like Japan, where they did a mass vaccination targeting school children, not adults, the death rate of the elderly went down.
Wow, that's interesting.
Because the total amount of virus in circulation went down. If this was November, it could have been a very different picture. A very different picture. So our feeling is we may, in fact, be faced with exactly the same thing or something even worse by November of this year. So we have a once in a lifetime opportunity to try and prevent it. And I think that we have no choice but to go forward with a pretty aggressive vaccine strategy. We don't know everything about influenza pathogenesis. We just don't. If we did, we'd have a much better surveillance system. But we don't. So in other words, we don't know what amino acid changes to look for that will tell us for sure, oh, this virus now is, this H5N1 virus is the one that's going to go from person to person. We don't have that level of understanding of the virus. Until we do, we need to be very cautious.